Next, we have ribosomes. These are going to be the sites of protein synthesis. Now, their name reflects their high content of one type of ribonucleic acid, which is called ribosomal RNA, or also called rRNA. But each one of these ribosomes also includes more than 50 proteins. So it's made up of rRNA and also a lot of proteins. Now, structurally, the ribosome is made up of two subunits. We have a large subunit and a small subunit. The small subunit is approximately half the size of the larger one. And these two subunits are made separately in the nucleus, and then they exit the nucleus separately, and then they come together in the cytoplasm. So these are made in the nucleus separate. Now, ribosomes can actually be found in a couple of different places. They can either be attached to other organelles, such as the outer surface of the nuclear membrane, the membrane on the nucleus, or they can also be attached to an extensively folded membrane called the endoplasmic reticulum. So I'll put here they can be attached to the nucleus, or they're also found on the endoplasmic reticulum. We'll just abbreviate that ER, but that's endoplasmic reticulum. Or they can also be free. This is just meaning that they are unattached to other structures in the cell. They're just kind of free floating in the cytosol. So you can either find them attached or free. Free ribosomes are used to synthesize proteins that are used in the cytosol. So the proteins that are made by ribosomes, just that are free floating, they are used in the cytosol themselves. There are also ribosomes located within the mitochondria. But these are just used to synthesize mitochondrial proteins, so proteins that are used only in the mitochondria. So next, we're going to talk about the endoplasmic reticulum. Now, this is a network of membranes in the form of flattened sacs or tubules, and it extends from the nuclear envelope. Remember, that's the membrane around the nucleus, and it projects throughout the cytoplasm. The endoplasmic reticulum, or also known as the ER, it's a very extensive network, and it makes up more than half of the membranous surfaces within the cytoplasm of most cells. Now, there are two types of ER within cells. There's going to be the rough ER, or the rough endoplasmic reticulum, and the smooth ER. So first off, the rough ER is going to be continuous with the nuclear envelope. And it's usually folded into a series of folded sacs. So we can kind of see these little fold structures here. And that's what the rough ER is going to look like there. The outer surface of the rough ER is also studded with ribosomes. So we can see those here, kind of blue in this picture. And these ribosomes are going to be the site for protein synthesis. Proteins that are synthesized by the ribosomes attached to the rough ER, they enter the spaces within the rough ER and they are processed and sorted. Okay, now the proteins that are made by ribosomes in the rough ER are going to be secretory proteins or membrane proteins, proteins that are put out onto the surface of the cell. These are going to be kind of stuck on the membrane of the cell. And then there's going to be a lot of other organellar proteins. Proteins that are used by other organelles. Okay, then we have the smooth ER, and this one extends from the rough ER and it really just forms a network of these membranous tubules. So you can kind of see here the rough ER is very flat and kind of folded, whereas the smooth ER kind of forms more of these little tubes. So it does look quite different from one another. Now, unlike the rough ER, the smooth ER does not have any ribosomes on the outside surface, but the smooth ER does contain unique enzymes that make it a lot more like functionally diverse than the rough ER. Now, because there's no ribosomes on it, the smooth ER does not synthesize proteins, but it does synthesize things like fatty acids, we'll say acids. Let me write that here. Fatty acids, also steroids. And these are going to be steroids like estrogens and testosterone. Next is the Golgi complex. This is also called the Golgi apparatus. We'll just put AKA, also known as. Now, this has a special function as well, because most of the proteins that are synthesized by the ribosomes attached to the rough ER are going to be transported to other regions of the cell, but the first step in this transportation is going to be the Golgi complex. So these are going to be proteins made by rough ER ribosomes are going to come to the Golgi apparatus. So this is going to be the first step after those proteins are made in the rough ER. They're going to come to the Golgi complex. Now the Golgi complex has a couple of functions. So what it mostly does is it's going to package proteins into nice little vesicles. We can see these here. And it's also going to modify proteins. So these are going to be kind of the main functions of the Golgi complex. Now the Golgi complex is made up of 3 to 20 of these cisternae. Now cisternae are going to be very small, flattened sacs that have these bulging ends, you can see here. Now, cisternae at the opposite ends of the Golgi complex will differ from each other in size and shape, and also in the enzymatic activity. So this would be the cis phase versus the trans phase. So this is where they enter in the cis phase on this side, and they exit here on the trans phase. So the cis phase versus the trans phase, like I said, they have a difference in size and shape and also different enzymes between them or within them. So the cis face is going to be this convex surface, and this is where it's going to face the rough ER, and it accepts the proteins that are synthesized by the ribosomes attached to the rough ER. This is where those vesicles that have proteins made by the rough ER ribosomes come in. And they will travel through all these middle cisternae. So they will move this way, traveling through all these cisternae. And as they travel through these folds in the Golgi complex, there's different enzymes and different structures within the Golgi complex that will modify these proteins, package them differently until they reach the trans phase. And then they are secreted out. And now that they are modified and packaged properly to go where they need to go. So here we can see the Golgi complex in a little bit more detail. We can see here the rough ER on the left side with the ribosomes attached to that rough ER. This little orange squiggle here we can see is a synthesized protein that's been synthesized by those rough ER ribosomes, and it gets put into a nice little transport vesicle. That transport vesicle will travel through the cytoplasm into the entry phase cisterna. So we can see number 3 here, that transport vesicle is endocytosed into the Golgi complex there. Now this little protein will travel through the medial cisternae. This is where carbohydrates are added, glycoproteins, lipids are going to be added to these proteins as they travel through the Golgi complex. And then the exit, or the trans phase, this is going to modify these molecules a little bit further and then sorts and packages them to transport them to their very specific destinations. So here we can see one is going to lysosomes. This one labeled number 8 is going to be sent to the plasma membrane. And then this one's going to be secreted over here. So these proteins are secreted, exocytosed from the cell because they are secretory proteins, proteins that are secreted out. Things like hormones would be secreted. Here, these proteins are built into the plasma membrane, kind of merged into the membrane. And this vesicle here, this transport vesicle, is going to stay within the cell. Next, we have lysosomes. These are going to be membrane enclosed vesicles that form from the Golgi complex. Remember, we just saw on the right side of that last diagram with the Golgi complex, those vesicles that stay within the membrane, these are going to be lysosomes. And they contain as many as 60 kinds of these very powerful digestive enzymes. And these enzymes break down really a wide variety of molecules once these lysosomes fuse with vesicles that are formed during endocytosis. Now, lysosomes mainly work to break down proteins that come to them in the form of vesicles. So they work to break down molecules in other vesicles when they fuse together with those other vesicles. Now, Lysozymal enzymes work best at acidic pHs. Now, within these membranes here, there's going to be active transport pumps. So these are going to be active transport pumps that are going to be constantly importing hydrogen ions. And this allows the interior of lysozymes to have a pH of approximately 5. So this is going to be very, very acidic. It's about 100 times more acidic than the pH of the cytosol, which is 7. So it means kind of like in this picture up here, we have a pH of 7, and within the lysosomes, we have a pH of 5. So very, very acidic. Now, pretty much the lysozymes kind of act like the trash bins of the cell. They are going to take in damaged or unneeded organelles, debris, or really anything else, any garbage that the cell has, and it's going to digest it completely. And then the lysozymes are going to release the usable molecules back into the cytosol. So it's going to things like glucose, amino acids, and fatty acids. These are all going to be broken down molecules that can be used by the cells for other things. Next, we have proteasomes. Now, as you can probably guess from the prefix proteal, these are going to break down proteins. But unlike lysosomes, which also break down proteins, proteasomes degrade proteins that are found free floating in the cytosol. Now, for example, proteins that are part of metabolic pathways, they need to be degraded after they have accomplished their function. Now, these protein destruction pathways, like what a proteasome does, plays a negative feedback into halting the pathway once the appropriate response has been achieved. So essentially, the proteasome is going to find proteins that have completed their function and break them down so that those proteins don't continually to try and complete their function over and over and over again when they don't need to. Once they're finished once they can be broken down, they are finished with their role. Proteasomes are really small and they chop up proteins into amino acids, which can then be used by the cell to be recycled into new proteins. Essentially, lysosomes and proteasomes just work as recyclers, essentially. I know I say garbage bins for lysosomes, but really it just breaks everything down into its smallest unit to be reused by the cell and recycled into other new fatty acids, new proteins, new carbohydrates. Now, the next organelle we're going to discuss is the mitochondria. This is known as the powerhouse of the cell, and this is because most of the energy in the form of ATP is going to be created in the mitochondria, and this is usually through aerobic production, aerobic respiration, which we will discuss later. Aerobic respiration is going to mean that it requires oxygen. Now, a cell may have as many as a few hundred or even several thousand mitochondria within it. Now, a little bit just on the anatomy of the mitochondria. It has an outer membrane and an inner membrane, so two membranes, and there's going to be this small fluid-filled space between those two membranes. There's going to be these really elaborate folds that we call cristae that provide a really enormous surface area for chemical reactions that are part of the aerobic phase of cellular respiration. And remember, these are going to be the reactions that produce most of a cell's ATP. Remember the ATP, this is going to be the energy for a cell. Now, the mitochondria also serves a really important role in apoptosis. This is also just another term for programmed cell death. Now, apoptosis can occur in response to many things. Could be destructive free radicals, damage to DNA within the cell, could be a lack of oxygen and nutrients, and all these reasons would result in certain chemicals being released from the mitochondria, and this creates a pore in the outer mitochondrial membrane. Now, one of these chemicals is known as cytochrome C. And generally, this is in the mitochondria pretty happily. It works in creating ATP. But if cytochrome C is released into the cytosol, it initiates this cascade of activating protein digesting enzymes that bring about apoptosis. So we're going to learn a lot about this a little bit later, but essentially we know that the mitochondria can essentially kill the cell if certain conditions are met. If certain things happen, like there's a lack of oxygen and nutrients, the mitochondria just kind of flips a switch and releases cytochrome C into the cytosol, and this brings about cell death or apoptosis. Now, a cool thing about the mitochondria is that it has its own set of DNA. This is called mitochondrial DNA. And this is DNA that's completely separate from the nucleus. Now, genes from the nucleus, these are going to be inherited from both your father and your mother, but mitochondrial DNA is only inherited from your mother. Now moving on to the nucleus. This is a spherical or sometimes oval shaped organelle. It's usually the most prominent feature of a cell. Now most cells will have just one single nucleus, though there are a couple exceptions. For example, red blood cells, they have no nucleus. And skeletal muscle cells and some other cell types, they will have multiple nuclei in each cell. Now there is a double membrane that encloses the nucleus. So you can kind of see the nuclear envelope here, a little bit of a zoom in. We have kind of this inner membrane here and an outer membrane here, and there are pores throughout that double membrane. Now these nuclear pores help to control the movement of substances between the nucleus and the cytoplasm. Small molecules and ions can move through the pore through passive diffusion. Remember, they'll have to move down their concentration gradient, like we discussed before. But most larger molecules, like RNAs and proteins, they can't pass directly through the pores. They actually have to pass through using active transport. So it's only usually small little ions and molecules that can directly pass through these nuclear pores. Now within the nucleus, we have the hereditary units called genes, which can be condensed into chromosomes. So here we have the DNA double helix, and this is going to have the instructions for all the genes that make up your whole body. So for example, we could have maybe from here to here on the DNA. This could hold the instructions for eye color, for example. So this DNA double helix, it's going to have linked nucleotides that are going to twist around histones, which are going to be proteins. And those histones are going to twist together and further twist and compact into nucleosomes, and you can kind of see they even compact and twist even more here and continue twisting until they kind of sort of resemble this bow tie. This bow tie structure is called a chromosome. Now, as you can see, because it's so compacted, there's going to be really a ton of DNA within these chromosomes. Now, DNA encodes for everything in the cells. Pretty much, DNA is the blueprint for all the proteins that a cell needs to survive. Now, in a process called gene expression, the gene's DNA is going to be used as the template for the synthesis of a very specific protein. So first, kind of step one, the DNA is going to be transcribed. Now, transcription essentially is a process where DNA is going to be rewritten into RNA. Specifically, DNA is going to be converted or rewritten into a specific type of RNA called mRNA, otherwise known as messenger RNA. Now, that copied mRNA molecule is going to be transported into the cytosol, or into the cytoplasm here, and it's going to be transported to a ribosome. Now, the RNA is going to attach to the ribosome, and this is where we have step 2, where the protein is going to be translated from that copy of DNA. So we'll go over transcription and translation in a little bit more detail here. So the first step being transcription. This is where DNA is going to be unwound. So we're going to kind of unwind that double helix, and the DNA is going to be copied into a different but more readable form. So this form is called mRNA, like I said. So it's going to be kind of converted or rewritten into mRNA. Remember, this is messenger RNA. And nucleotides, which make up DNA and RNA, are used to transcribe the DNA code into mRNA. Now, there's a lot of modification that occurs on this mRNA molecule, as the entire transcribed portion doesn't actually code all for a protein. So when a long portion, we won't actually go into too much detail about this, but say there's a long portion of DNA that is transcribed into mRNA. So we'll say this is going to be the DNA portion, and it's going to be converted into mRNA. Now, there's going to be sections within here that are called introns. So maybe from here to here, this is an intron, and which is different color here. Maybe from here to here, this is an exon. Now, exons, these are going to be the portions that actually code for proteins. So this is going to be the actual protein code. And introns, these are kind of just filler code. Doesn't actually code for any particular protein, just kind of filler RNA. So it doesn't actually code for proteins that we know of. It could do something else or have another function, but we kind of don't think that it codes for any type of proteins. Now, there are very specific enzymes that will cut the introns out of the mRNA so that we're just left with that functional protein coding code that's all left over. So that we cut out the introns, we're just left with exons. Now, all of this processing all occurs in the nucleus before the mRNA is sent out of the cytosol. Now, this is kind of just a brief overview. You don't need to remember all these specifics, but just know that the exons in mRNA code for proteins, and mRNA is just going to be that more readable version of the DNA code. Now, the second step in protein synthesis is going to be translation. Now, translation occurs in the cytosol. This is after the mRNA has been transcribed from DNA, and we've also modified that mRNA so that we're removing all of those introns. We've sent it out of the nucleus into the cytosol, and then this is where we get to translation, the actual protein synthesis part of this entire process. So this is where the mRNA comes to ribosomes, and the ribosome will kind of read it, and it will tell the ribosome which protein to make. So here we have this long mRNA segment. We can see right here is labeled, and it first is going to attach to the small subunit of a ribosome. And then the large subunit is going to join together, and essentially the large and small subunits will read the mRNA, and it'll tell us, or it'll tell the ribosome rather, which amino acids to bring in. The ribosomes will use these other type of RNA, which is called tRNA. This is the transport RNA. And tRNA is going to be the actual structure that brings amino acids to the ribosome for building proteins according to the mRNA strand. So say that the mRNA has a code like AUG. AUG, for example, would code for the amino acid methionine. And so pretty much the mRNA is going to tell the ribosomes to bring methionine because we're coding for methionine. This is what the instructions say. Instructions say methionine is going to be the first amino acid in the sequence. And so the ribosomes will talk to tRNA and tell tRNA, hey, bring methionine. That's going to be the first instruction that this mRNA is telling me to build. And so it will continue in that process until we build more and more amino acids, kind of making this chain. That's kind of the gist of it. We don't need to know entire process of transcription. The main thing is that we need to know is that mRNA brings the instructions to the ribosomes, and the ribosomes will read the instructions and tell tRNA which amino acids to bring in order to build those amino acids into a chain, creating the protein that mRNA codes for. So we've kind of gone over all the organelles in the cell, or at least all the organelles in the cell that you need to know for this class. So now we're going to briefly go over cell division. So mitosis occurs when somatic or body cells need to duplicate. Somatic cells is just another word for body cells. So we're going to talk about mitosis. So human cells contain 23 pairs of chromosomes. And so this is a total of 46 chromosomes completely. And some cells will divide more than others. When a cell reproduces, it must duplicate all of its chromosomes to pass on its genes to the next generation of cells. So cell division is really just that process in which cells reproduce. So we can kind of see the cycle here on this picture on the right. Now, most of the cell cycle consists of what is called interphase. This is in the light purple color here. And interphase is really just the preparation for cell division, but it's not dividing yet. So here, this is really just division preparation. So interphase is kind of more divided into 3 different phases. This would be the G1 phase, the S phase, and the G2 phase. Pretty much G1 phase is where the organelles and cytosolic components are duplicated. G2 phase, this is where pretty much like the enzymes and other proteins are going to be synthesized. And then our S phase is going to be our major phase where DNA is all replicated. Mitosis down here, or the mitotic phase, this is when the cell is actually dividing. So this is going to be the dividing phase, and it is broken up into, we call it PMAT, P-M-A-T. This is also known as prophase, metaphase, anaphase, and telophase. Now, cells will always start into mitosis, kind of right here. This is where they're always going to start as a diploid cell. This term diploid means that the cell contains 2 complete sets of paired chromosomes, or duplicated chromosomes. I'll just put dup-chrome for duplicated chromosomes. So this is when all of their DNA has been replicated, and they're coming into the beginning of metaphase with completely like 2 sets of everything. Then they're going to go through this mitosis where we have 1 cell is going to divide into 2 cells. And so we're going to end up with 2 haploid cells. Haploid is a term where it means that these cells have a single set of unpaired chromosomes. We also term diploid 2N, where we have 2 sets of genes, and haploid just being a single N, which means it is a single set of genes. So here's just showing we have interphase being broken up into G1, S, and G2. Interphase, we pretty much have all of this chromatin. This is just the non-condensed version. This is when DNA is all just kind of spaghetti looking. This is kind of non-condensed DNA. So this is when DNA hasn't been completely coiled up and turned into those chromosomes. It's just kind of uncoiled. It kind of fills the entire space within the nucleus. Now we get into mitosis. Now, mitosis is really an exact partitioning of genetic information into 2 identical cells. So there are really 4 stages of mitosis, remember that PMAT, but this is a continuous process where each phase kind of seamlessly merges into the next. So remember, during that interphase, we had this very spaghetti-like, non-condensed DNA material, but during prophase, this first phase of mitosis, the chromatin fibers are going to really condense and shorten. And they're going to shorten into chromosomes that are going to be very visible under light microscopes. So chromatin, you can't really see it very well under a microscope because it's all just very spread out and isn't really condensed enough to see. But once it's condensed into chromosomes during prophase, we can actually see them under a microscope. Now, because that longitudinal DNA replication took place during the S phase of interphase, each prophase chromosome consists of a pair of identical strands that are called chromatids. So each chromosome consists of 2 chromatids that are joined at a centromere. This is going to be this constricted region that holds the chromatid pairs together. So here, I'll just draw a little picture here for you. We have one chromatid here. I'll draw in a different color, but these chromatids are going to be identical to each other, kind of. And in the center here, this is where we have the centromere, this constricted region holding those chromatids together. And just outside this centromere, kind of like right here, we have this protein complex that is known as a kinetochore. You can see this is labeled here. Now, a little bit later in prophase, kind of here we have early prophase and late prophase. This is when tubulins in the centrosomes are going to form the mitotic spindle. So here we have those centrosomes in green. Kind of talked about them a little bit before. We have these mitotic spindles growing off of those centrosomes. Now those mitotic spindles are actually going to attach to the kinetochores of the chromosomes. So we can see this here. We have one mitotic spindle coming here and attaching to the kinetochore there, another one here attaching to this kinetochore, so on and so forth. Now, kind of also what happens in late prophase is that we have the nucleus disappear, or the nuclear envelope disappear as well, and also the nucleolus disappears. So we can kind of see in this left picture, we have that nuclear envelope around here. Still starting to break down in this picture, but here in late prophase on the right side, we have no nuclear envelope. Now that next phase of mitosis being metaphase, this is where the microtubules of the mitotic spindle will align the centromeres of the chromatid pairs at the exact center of the mitotic spindle. So we have mitotic spindle going from here to here, and then here at the exact center, we call that the metaphase plate. So the chromosomes will be lined up at the metaphase plate during metaphase. Now during anaphase, the centromeres are going to split, separating the 2 members of each chromatid pair. So see you have one chromatid here and one chromatid here. They're going to be pulled to opposite poles of the cell. Now once they are separated, the chromatids are actually now each individually called chromosomes. So you can see this is labeled here. So this one's a little backwards. It has early anaphase on the right, late anaphase on the left here. But those chromatids are going to get separated, and once they are their own unique structure, they're called chromosomes. And as they're pulled apart from each other, the cell starts to kind of divide itself, kind of separate itself into the beginning of 2 cells, and this is by a cleavage furrow. So it's kind of starting to constrict just a little bit in the middle here, the beginning stages of separating into 2 distinct cells. Now the final stage of mitosis, which is telophase, this phase begins after chromosomal movement has stopped. The chromosomes are now in their respective poles, and they're going to uncoil and revert back to that thread-like chromatin form. So they're going to regain their chromatin appearance. We're also going to see a new nuclear envelope or a new nuclear membrane form on either side. So we can see this here on both sides. And these mitotic spindles will start to break up. We get also to get a little bit deeper of a cleavage furrow here. But even at the end of telophase, the complete separation of these two cells is not finished yet. This is where we get to cytokinesis. So cytokinesis, this is a term given to the division of the cell's cytoplasm and organelles into two identical cells. So this entire process of cytokinesis usually begins in late anaphase with that beginning formation of the cleavage furrow, and cytokinesis is completed after telophase. So here we can see anaphase. This is when we're starting to have cytokinesis begin with that beginning of the cleavage furrow. In telophase, a little bit more of a cleavage furrow. But then after telophase, cytokinesis is finished when these cells are completely separate. So we can kind of have this process be summarized like this. So we have the G1 phase. G1 goes into S phase. S phase goes into the G2 phase. G2 phase goes into mitosis. And mitosis go into cytokinesis. But again, some of cytokinesis occurs during mitosis as well. Here's a nice summarizing table of all the events of a somatic cell cycle. So again, somatic cell just means all of your body cells. Just includes all the phases we discussed and the activities of those phases. Now there are three possible destinies for every cell. They can remain alive and just function without dividing. They can grow and divide, or they can die. So these are going to be our essentially three fates that can happen to any cell. Okay, we are going to very briefly go over meiosis, which is a little bit different from mitosis, but just a few differences between them. Now, reproductive cells or gametes will go through meiosis. This is also known as reproductive cell division. And reproductive cell division occurs in the gonads. So this is in the ovaries and the testes. Reproductive cell division produces gametes, so these are sperm and ovum, and each of these contain a single set of 23 chromosomes. Fertilization of the two gametes, one sperm and one ovum, will restore the diploid number of chromosomes. Now, unlike mitosis, which is complete after a single round, meiosis, on the other hand, has two successive stages. Meiosis 1 and meiosis 2. So meiosis 1 begins once chromosomal replication is complete, and it consists of 4 phases. Prophase 1, metaphase 1, anaphase 1, and telophase 1. Now, there's this process called crossing over that occurs during prophase 1, and this allows there to be an exchange of genes between chromatids of homologous chromosomes. So you can kind of see right here that we kind of get this crossing over, and then we get two completely different chromosomes here. So the resulting cells from this crossing over allows the cells to be completely genetically unlike each other and completely genetically unlike the starting cell that produced them. Homologous chromosomes, or the same paired chromosomes, will pair up along the midline in prophase 1. And then they go through this crossing over here. And then they'll go into anaphase. They go through metaphase first, but this is just when they kind of pair up along the metaphase plate. And then in anaphase, the members of each homologous pair of chromosomes will separate, and they're going to be pulled to each pole. So you can kind of see here we have these kind of mixed up chromosomes now with a little bit of blue and a little bit of red on them. So they're very different from the original chromosomes up here in prophase. So this kind of separation that we see here in anaphase 1 is actually different from mitosis. Remember, in mitosis, the centromeres will split in anaphase and the sister chromatids separate. So here we have full chromosomes, full bow tie chromosomes like this, that will actually be pulled to each individual pole. So this one goes that way and that one gets pulled that way. Instead, in mitosis, we would have this chromosome, or chromatid, I guess in this case, get pulled that way and this way. This was in mitosis. So it's a little bit different between meiosis 1 and mitosis, even though they both still occur in anaphase, but anaphase is a little bit different between the two. So the net effect of meiosis 1, which is just going to be this one, this is going to be resulting in every cell containing a haploid number of chromosomes, because it contains only one member of each pair of the homologous chromosomes that are present in the starting cell. So we're going to end here with 2 haploid cells. Meiosis 2 is essentially the same steps, and it's essentially the same, but meiosis 1 starts with a diploid cell and ends with 2 haploid cells, and meiosis 2 starts with 2 haploid cells in prophase 2, which will each divide and result in 4 haploid gametes that are genetically different from the original cell. So here we have four haploid gametes resulting at the end of meiosis 2. Also, one other thing to note is that crossing over that we saw earlier only occurs in prophase of meiosis 1. So it only happens in prophase 1. We don't see that same crossing over occur in prophase 2. Now, as we age, there are a few things that happen to our cells. Just very briefly, they start to deteriorate in their functioning. So they can't function as well as younger cells. The actual number of body cells decreases. It's because the body cells will die as they get older and they're not replaced by newer cells. And also the integrity of the extracellular components of tissues deteriorates. We also get free radicals more as we age. So a free radical is an uncharged molecule. Typically, they are very reactive as they have unpaired valence electrons. So in the body, these can actually be quite destructive. And that's going to be the end of chapter 3. This is chapter 4, and we're going to be looking at the tissue level of organization. So in this chapter, we're going to be looking about the various different types of tissues. We're going to be specifically looking at epithelial, connective, muscular, and nervous tissue. We're also going to look at the structure and function of membranes, and then look a little bit just into tissue repair. So the 4 tissue types in our body are epithelial, the left here, left top. Then we have connective, which is top right, muscular, which is bottom left here, nervous down here. So these are going to be our 4 tissue types, and we will talk about each of them individually. So first off, we're going to be looking at the differences between epithelial versus connective tissue. So just look at these 2 here. The one on the left is epithelial tissue, and the one on the right is connective tissue. Now the first obvious difference that you can see here is the number of cells in relation to the extracellular matrix. So in the epithelial tissue on the left, there are many cells like very tightly packed together with little to even no extracellular matrix. Whereas the connective tissue on the right, there is a large amount of extracellular matrix separating the cells that are usually very widely scattered. So we only see a couple of nuclei here and a lot of extracellular matrix around them, whereas on the left here, there is a ton of nuclei kind of everywhere. Secondly, is that epithelial tissue has no blood vessels. So we're also going to add that here. No blood vessels. Whereas most connective tissue has quite significant networks of blood vessels. Now, because epithelial tissue lacks blood vessels, and epithelial tissue, we'll find out a little bit later, but this forms surfaces. So you're always going to have one end of epithelial tissue on this side, in this picture, is going to be a surface. So it's going to pretty much be exposed to the exterior. So this could be skin, this could be the inside of your intestines, which is going to be technically exposed to the exterior. And so this is going to form a surface. So since there's not many blood vessels and epithelial tissues form surfaces, it's always found immediately adjacent to very blood vessel rich connective tissue, and this allows epithelial tissue to make the exchanges necessary with blood in order to deliver oxygen and nutrients and also remove wastes. These are all very critical processes for survival and functioning of tissues. So first we're going to go into a little bit more detail about epithelial tissue. So epithelial tissue has 5 distinguishing characteristics. First one is polarity. So all epithelia have 2 surfaces that are quite different from one another. So these are going to be the apical surface and the basal surface. Essentially, it is going to be the top side and the bottom side of the tissue. The apical surface is not going to be attached to anything, and so it's either going to be exposed to the outside of the body or exposed to the internal cavity of an organ. The basal surface, on the other hand, is going to be attached to the connective tissue below. The next characteristic is going to be specialized contacts. So except for glandular epithelia, epithelial cells will fit very closely together to form continuous sheets of cells. They're generally held together by tight junctions. Remember, we talked a little bit about tight junctions in last chapter, and these tight junctions really prevent substances from leaking through the spaces between cells. Next characteristic is that they are supported by connective tissue. So all epithelial tissues will rest up and are supported by connective tissue. And between the epithelial layer and the connective tissue layer is what is called the basement membrane. And the basement membrane works to resist tearing and stretching, and it kind of reinforces the epithelial layer. But that basement membrane is going to be also separating the epithelial layer from the connective tissue layer. The next characteristic is regeneration. So epithelium has the ability really to regenerate extensively. So since epithelial tissues are going to be subject to a lot of friction, the surface cells rub off pretty frequently. Now, as long as epithelial cells receive enough nutrition, they're going to be replaced, or the lost cells are going to be replaced by cell division pretty quickly. Lastly is that epithelial tissue is avascular, meaning it does not have a blood supply, but it does have a nerve supply. So epithelial tissue has no blood vessels within it, meaning it's avascular, no vessels, but it is supplied with nerve fibers. So these cells are going to be nourished by the blood vessels in underlying tissues. Now, the various surfaces of epithelial cells will differ in structure, and some of them have specialized functions. But here we just kind of have a general epithelial cell little section. So here we can see the apical surface. This is, remember, going to face the body surface or the body cavity, or even the lumen, the interior space of an internal organ. This apical surface could also open to a tubular duct as well that receives cell secretions. So this could open to the body surface, body cavity, lumen of an organ, or a tubular duct. And we'll go over ducts a little bit later as well. Then we have the lateral surfaces. And these are going to face adjacent cells, and they may connect by several junctions between them. And then we have the basal surface down here. This is going to be the opposite side of the apical surface. And the basal surface is the deepest layer of epithelial cells, and it adheres to extracellular materials, like the basement membrane in this case. So here we can also see that the basement membrane separates the epithelium from the connective tissue. Now, covering and lining epithelium forms the outer covering of skin and some intestinal organs, like I've discussed earlier. It also forms the inner lining of blood vessels and ducts and body cavities, even the interior of respiratory, digestive, urinary, and reproductive systems. Now, there's a couple different ways that we classify epithelial tissue. This is going to be both on the arrangement of layers and also on the shape of the cells. So the layers could be simple, which is just one single layer where every cell has an apical surface and every cell has a basal surface that touches the basement membrane. Then there is stratified, where there is one layer of cells that all touch the basement membrane, but they don't have an apical surface. There's going to be one layer of cells that touches the apical surface, and then just lots in between. So stratified just means multi-layered. And pseudostratified, this is where every cell touches the basement membrane, but not every cell has an apical surface. Cell shape, squamous just means flat. Cuboidal is cube-shaped, and columnar is column-shaped. Here's a nice little table that kind of shows the different shapes and arrangements that can go together. So simple arrangement, so that, remember that single layer, they could be either squamous, cuboidal, or columnar. Pseudostratified is the only one that can only be columnar in shape. And then stratified could be either shape as well. So first we'll talk about glandular epithelium. Now the function of glandular epithelium is secretion. Now this is accomplished by glandular cells that often lie in clusters deep to the covering and lining epithelium. Now a gland may consist of a single cell or a group of cells that secrete substances into ducts. Now, all glands in the body are classified as either endocrine or exocrine. So the first one we have here on this page are the endocrine glands. So endocrine glands will secrete hormones, and they will secrete hormones into the interstitial fluid. They will not secrete hormones directly into the bloodstream, but they secrete hormones into the interstitial fluid around them, and then those hormones will diffuse directly into the bloodstream, and they do so without flowing through a duct. Endocrine glands really have far-reaching effects, and this is because they are distributed throughout the body, and these hormones can travel through the bloodstream. The thyroid gland here is an example of an endocrine gland. Exocrine glands, these are glands that secrete their products into ducts, and those ducts will empty onto the surface of a covering or lining epithelium, for example, like a skin surface or the lumen of a hollow organ. Now, these secretions have very limited effects, and some of them would be harmful if they entered into the bloodstream. Now, some glands of the body, like the pancreas, ovaries, and testes, these are going to be determined as mixed glands. So mixed glands like the pancreas, ovaries, and the testes. Mixed glands mean that they contain both endocrine and exocrine tissue. Now, glands come in several different structures and functions. They could be unicellular or single-celled glands. They also could be multicellular, and these are going to be most of the ones we're going to be talking about for this class. These are ones that are composed of many cells, and they form either a distinctive microscopic structure or macroscopic organ. So examples of multicellular glands would be sweat glands, oil glands, and salivary glands. So here are some of the shapes that we find our multicellular exocrine glands are common. Could be just a simple tubular or simple branched tubular. I'll also see simple coiled or simple acinar or bulb shaped, or a simple branched acinar. And then we kind of get a little bit more complicated here with these compound tubular, compound acinar, or compound tubuloacinar. Ones that we're mostly going to talk about are going to be on this top row, but you can kind of just keep this slide in mind as we talk about some specific types of glands and how they may look. So let's look at the functional classification of different glandular epithelium. So we have three different functional classes: merocrine, apocrine, and holocrine glands. So first here we have the merocrine glands. Now, secretions of the merocrine glands are synthesized in ribosomes that are attached to the rough ER here. Then they are processed, sorted, and packaged by the Golgi complex, and then they're released from the cell in secretory vesicles, and this is by exocytosis. Sweat glands and salivary glands are going to be this type of functional classification. So we'll put this here. Sweat glands, salivary glands. These are both going to be types of merocrine glands. Next, we have apocrine glands. It's going to be our middle one here. Now, these are going to accumulate their secretory product at the apical surface of the secreting cells, and then that portion is pinched off of the cell. This is going to be pinched off by exocytosis from the rest of the cell, and this is how that secretion is released. The cell is then going to repair itself, and then they can repeat the process all over again. Now, this is the mechanism of secretion of milk fats in the mammary glands. Then we have holocrine secretion. Now, cells that undergo holocrine secretion will accumulate a secretory product in their cytosol. And then as the cell matures, it will rupture and the entire cell becomes the secretory cell, or the secretory product rather. Now, since that whole cell ruptures, the secretion contains large amounts of lipids from the plasma membrane and cellular membranes. So this occurs in the sebaceous glands or the oil glands in the skin. So now we're going to move on and talk about connective tissue. Now, connective tissue is one of the most abundant and widely distributed tissues in the body, and it does have several functions. It functions to bind together, it supports, it strengthens other body tissues, it protects and insulates internal organs, and it serves as a transport system within the body. For example, blood is actually a fluid connective tissue. It also compartmentalizes structures like skeletal muscle. It's also the primary location of stored energy reserves like adipose or fat tissue. And it's the main source of immune responses. So lots of important functions of connective tissue. Now there are two basic elements in all connective tissue. These are the cells and the extracellular matrix, or pretty much everything else around the cells. Now the extracellular matrix, this is going to consist of protein fibers and ground substance. Now, ground substance, this is essentially going to be the material between the cells and the fibers. The extracellular fibers are secreted by the connective tissue cells, and they account for many of the functional properties of the tissue, in addition to controlling the surrounding watery environment by very specific molecules. So here we can kind of see a lot of those cells. We can see that ground substance, that material around all the cells and the fibers, and we can see several of these different types of fibers in connective tissue. So we can see reticular fibers here, elastic fibers, and collagen fibers here. Now, connective tissue is highly vascularized, so we can see this blood vessel here. So a lot of blood vessels within connective tissue. Remember, this is very much unlike epithelial tissue. There are some exceptions though. Cartilage, here I'll put avascular. Avascular connective tissue would include cartilage. It is completely avascular, and tendons have a very small blood supply. They're not completely avascular, but they have a very minimal blood supply. So except for cartilage, connective tissue is going to be very much supplied with nerves as well. We can see these nerves in this picture. These are going to be these yellow lines throughout. These are going to be your nerves here. So connective tissue and epithelial tissue both are very highly supplied with nerves. In this picture, you can also see several different cells that we will talk about and you'll learn about throughout the class, including macrophages, which pretty much clean up debris by phagocytosis. Adipocytes, these are going to be fat storage cells. And then fibroblasts, these secrete fibers and ground substance. And then we also have a lot of immune cells here too, eosinophils, neutrophils, and plasma cells too. Mast cells are also kind of in the immune system section as well, but lots of different cells that we find within the connective tissue. So like I said, the extracellular matrix is made up of fibers and ground substance. And the ground substance is going to be that component of connective tissue between the cells and fibers. Now, ground substance could be fluid, could be semi-fluid, gelatinous, or even calcified, like we see in bones that we see later. Calcified ground substance makes up a lot of bone. Ground substance is going to support cells. It binds them together, stores water. It also provides a medium for exchange of substances between the blood and the cells. And it really plays an active role in how tissues develop. It helps cells to migrate and proliferate, change shape, and even kind of controls how cells carry out their metabolic function. This is a word that will come up later. Proliferate. This essentially means to grow and divide. Proliferate means to create more cells. So maybe like division and creating more cells. So let's discuss the different types of fibers within the connective tissue extracellular matrix. First, we have collagen fibers. These are very strong and resist pulling forces or tension. So resist pulling forces, but they are not really stiff or rigid. So this does allow for tissue flexibility. Now, the properties of collagen fibers will vary from tissue to tissue, and they're not completely consistent though. Now, chemically, collagen fibers consist of the protein collagen. It's in the name. And collagen is the most abundant protein in your body. It does represent about 25% of all the proteins in your body. Now, collagen fibers are found in most types of connective tissues, especially in bone and cartilage and tendons, which attach muscle to bone, and also ligaments, which attach bone to bone. So tendons attach muscle to bone, and ligaments will attach bone to bone. Okay, next we have elastic fibers. Now, elastic fibers are going to be smaller in diameter than collagen fibers, and they will branch and join together to form a fibrous network within connective tissue. Elastic fiber consists of molecules of the protein elastin, and elastin is going to be surrounded by a glycoprotein named fibrillin. And the fibrillin really adds a lot of strength and stability. Elastin fibers or elastic fibers are really unique and have a very unique molecular structure, and this allows them to be stretched to 150% of their relaxed length without breaking. Elastic fibers are found in skin, blood vessel walls, and in lung tissue. Okay. Then, lastly, we have the reticular fibers. Now, reticular fibers consist of collagen arranged in really fine bundles with a coating of glycoprotein. And reticular fibers provide support to the walls of blood vessels, and they form a network around the cells in some tissues. So they're found in areolar connective tissue, adipose tissue, nerve fibers, and in smooth muscle tissue as well. Now, just like collagen fibers, the reticular fibers provide strength and support and are plentiful in reticular connective tissue, and this forms the stroma of many soft organs. Stroma is the supporting framework. So reticular fibers will actually form the stroma in a lot of soft organs like the spleen or the lymph nodes. Reticular fibers also help to form the basement membrane. Now there are two major classification groups of connective tissue. These are embryonic and mature. So first off, very briefly about embryonic. We won't go over this one too much, but there are two types of embryonic connective tissue, mesenchyme and mucus. Mesenchyme is present primarily in the embryo and the fetus, but we do see mucus connective tissue later on in life as well. And then we have mature connective tissue. This is present in the newborn, and these cells are derived from the mesenchyme cells. Loose and dense connective tissue is just what you would think. They are loosely and densely arranged between cells. Now, types of the loose connective tissue are areolar connective tissue, adipose tissue, and reticular connective tissue. Now, dense connective tissue have considerably fewer cells than loose connective tissue, and we're going to find dense connective tissue in places like tendons and ligaments. Cartilage, this consists of a dense network of collagen fibers that are firmly embedded in chondroitin sulfate. Now, chondroitin sulfate is a gel-like component of the ground substance. Now, cartilage can endure considerably more stress than loose and dense connective tissue, and this is due to the collagen fibers and also the chondroitin sulfate, which adds a lot of resilience. Bone tissue. This is a connective tissue that makes up the skeletal system. Now, bones are organs that are made up of several different tissues, including bone or osseous tissue, the periosteum, red and yellow bone marrow, and endosteum, which is going to be a membrane that lines the space within bone and stores yellow bone marrow. We will go over the different structures of a typical bone when we talk about the skeletal system, so don't need to remember any of this, not much detail until we go over that chapter. So bone can be classified as either compact or spongy. And this depends on how its extracellular matrix and cells are organized. And like I said, we're going to cover all these differences between the different types of bones when we look at the skeletal system. Lastly, blood is also considered a connective tissue, but it's a liquid connective tissue. So it has a liquid extracellular matrix called blood plasma. This is the liquid extracellular matrix, and it also has formed elements. That's supposed to say formed elements there. Formed elements in the blood, these are going to be things like red blood cells, white blood cells, and platelets. And we'll go over all of the aspects of blood when we talk about the circulatory system. So that's a whole nother chapter as well. Now let's move on to talking about muscle tissue. Now, muscle tissue consists of elongated cells, which are called muscle fibers. They're also called myocytes. So we call them kind of both interchangeably. So myo, prefix meaning muscle, site meaning cell. And myocytes or muscle fibers are going to be the cells that make up the complete muscle that we know of. So these muscle fibers can use ATP to generate force. Now, based on their location, certain structural and functional features, muscular tissue is classified into 3 different types. So these are skeletal muscle, cardiac muscle, and smooth muscle. Now we'll talk about these a little bit more in detail when we go over the muscular system as well. And then we have nervous tissue. Now nerves, they can convert stimuli, mechanical stimuli, like being touched, for example, into electrical signals called action potentials. So the neuron is made up of 3 parts: the cell body, the dendrites, and the axons. So the cell body, this is where the organelles and the nucleus reside. Dendrites, these are going to be tapering, very highly branched and usually short extensions. These dendrites are going to be the major receiving or the major input portion of the neuron. And then the axon, which is a single thin cylindrical process that may be very long. And this is going to be the output of the neuron. And this is where nerve impulses are conducted towards other neurons or other tissues. This electrical signal goes down the axon. Within nervous tissue, we also see neuroglia. Now, neuroglia do not conduct nerve or action potentials. They're pretty much the support for nerves. They act as mechanical support, but also like metabolic support, giving them nutrients, removing waste, that kind of stuff. They're kind of like the stuffing around nerves. They help to, like I said, bring nutrients, take away waste, support the neurons in other ways as well. Okay, about membranes. So there are 2 types of membranes: epithelial membranes and synovial membranes. So first off, let's briefly talk about mucous membranes. So mucous membrane lines a body cavity that opens directly to the exterior. It's going to be the main definition of a mucous membrane. So places where we're going to have mucous membranes would be like the entire digestive tract, the respiratory and reproductive tracts, and much of the urinary tract. So they consist of a layer of epithelium and an underlying layer of connective tissue. Mucous membranes are really an important factor in the human body's defenses, because it's that barrier that microbes and other pathogens have difficulty penetrating through. Next, we have serous membranes. Now, serous membranes line a body cavity that does not open directly to the exterior. So, these would be places like the thoracic or abdominal cavities. Serous membranes cover the organs that are within that cavity. So remember that serous organs have 2 layers. We learned about that in the first chapter. They have a layer that attaches to and lines the cavity wall. That's going to be the parietal layer. And then they also have that layer that covers and adheres to the organs within the cavity itself, which is the visceral layer. Then we have cutaneous membranes. This type of membrane essentially is just known as skin. This covers the entire surface of the body and consists of a superficial portion called the epidermis and a deeper portion called the dermis. And we'll cover this entire cutaneous membrane or skin in a lot more detail when we talk about the integumentary system. Then we have synovial membranes. These line the cavities of freely movable joints, also known as joint cavities. Now, just like serous membranes, the synovial membranes line structures that do not open to the exterior, but unlike mucous, serous, and cutaneous membranes, synovial membranes lack epithelium, and so they are not an epithelial membrane, which is kind of why they get their own section here. Here we can see an example of a mucous membrane, remember, lining body cavities that are open to the outside, to the exterior. So this would be in the small intestine. Here we have a serous membrane lining cavities that don't open directly to the outside. It's going to be on the outside of the lungs, or I guess in the thoracic cavity. This lines the part of the lungs that attach to the rest of the thoracic cavity here. But within the lungs, on the interior, we're going to have a mucous membrane. Here we have the cutaneous membrane, or just skin. The entire skin is going to be the only cutaneous membrane, with a superficial epidermis and a deeper dermis covering the surface of the whole body. And here we have a synovial membrane. Synovial membrane, this is going to be a small finger joint. So here we have that synovial cavity with that membrane lining the cavity there. Now, just really briefly, last thing we're going to talk about is a little bit of tissue repair and aging. So tissue repair, this is going to be the process of replacing old, damaged, worn out cells or even dead cells. And epithelium has a really good repair process. They can repair and replace cells really rather quickly. And this is done by the division of stem cells near the basement membrane or by these undifferentiated cells. Connective tissue, on the other hand, they have some ability to repair, but not as much as epithelial tissue for sure, but they do have a little bit of ability there. Muscle cells also are very, very limited in their ability to repair and replace cells. And nervous cells, some can repair and some can't. It really depends on the neuroglia surrounding them and the types of cells that are supporting the nerves. And we'll discuss that a lot when we talk about the nervous system, and we'll kind of discuss which cells can be repaired and which can't within the nervous system. Aging is another thing that happens normally, which is a normal process. And so generally younger bodies live in a better nutritional state and have better blood supply to tissues, and also have a much faster metabolic rate. And so this all contributes to how quickly your tissues can repair and replace cells, and also how quickly your cells just function normally. So because of this, aging, getting older, really slows that process of tissue repair. That's going to be the end of chapter 4.